Hello everyone, my name is Paris Zhang, and for the past year and a half I have been working on this project in conjunction with the Louisiana Cancer Research Center in downtown New Orleans and Dr. Heitao Zhang's lab on assessing the implications of certain hotspot mutations in the androgen receptor in the cells of prostate cancer patients. I want to be the first to point out that I recognize the irony that my presentation for the Newcomb Scholars program, with its dedicated ethos to feminism, is on prostate cancer, a disease whose biological imperative is founded upon male biology and hormones, but I think it's important to recognize also that men are not the only sufferers of this disease. While prostate cancer is the second leading cause of cancer death in men in the US, its prevalence and fatality is an extremely heavy burden on the informal caregivers who tend to be spouses and daughters. A study by the University of Washington estimated that within 1 year of diagnosis, partner caregivers' average working hours decreased from 14 to 10.9 hours a week, offset by an increase annually of 66 hours of informal caregiving, plus an extra 76 hours a year to household chores. This study excludes the mental and emotional costs among partner caregivers. Thus, research in the field of prostate cancer will improve the quality of life of both male patients and the females surrounding them that are unwittingly often implicated. In this presentation, I'm going to first outline the mechanism and the role of the androgen receptor in the cell, then its role in prostate cancer, which will explain why androgen deprivation therapy is traditionally used to initially treat the cancer. Then I'll go on to introduce the two hotspot mutations we have identified in our lab as being potential cancer drivers and explain the significance of data from the lab and from data mining. So looking at this diagram, we can see that the androgen receptor, or AR, gene lies on the X chromosome. The AR gene is expressed in a variety of tissues in the body, and consequently, androgens have been linked to affecting muscle, bone, adipose tissue, as well as, of course, the prostate tissue and the reproductive system. This gene codes for a protein of a total of 919 amino acids, and this full-length protein consists of three major functional domains. These are the N-terminal transcriptional regulation domain, the DNA binding domain, and the ligand binding domain. We are most interested in the ligand binding domain as this is the part of the protein that the androgens interact with and bind to, and it is also the site of our two studied mutations called androgen receptor W742C mutation and the ART878A mutation. A quick side note, the mutation nomenclature here refers to first the letter that represents the original amino acid. The three numbers denote the position or location of the mutation, and the final letter stands for the change in the amino acid that classifies the protein as being mutated. For example, in ARW742C, the W stands for tryptophan, the wild-type amino acid, which has mutated into cysteine, another amino acid, and it is located on the 742 position, which locates it on the ligand binding domain. Androgens are the hormones responsible for the masculinization of male body tissues. You've probably heard of the main circulating androgen testosterone, which can also be converted via 5-alpha reductase into another androgen called DHT or dihydrotestosterone, but there are also other types of androgens. In a cell, the androgen receptor is responsible for the direct regulation of gene transcription, which is to say that these receptors allow the body to respond appropriately to the circulating hormones in the body. When the androgen binds to the androgen receptor, a conformational change occurs that activates a pathway of reactions. The diagram shows the pathway step by step, but essentially the resulting androgen receptor complex enters the cell nucleus and binds the DNA, which allows it to regulate the activity of certain genes. In a normally functioning cell, the androgen receptor turns genes on or off as necessary, which thus helps direct the development of male sexual characteristics. The following graphics represent a three-dimensional viewing of the androgen receptor and in particular demonstrate where the ligand is situated within the crystal structure of the ligand binding domain. In prostate cancer, the role of AR is generally maintained. For example, it is still responsible for the synthesis of prostate-specific antigens and the regulation of lipid metabolism, but it's also acquiring new responsibilities that promote the growth of the cancer, and much of this is still unknown. As a result, the current primary treatment for metastatic prostate cancer is androgen deprivation therapy, or ADT, which essentially removes the driver of this growth, as without androgens, the pathway of the androgen receptor complex into the nucleus that enacts on the regulation of gene transcription and growth cannot occur. The image on the left shows the AR with androgen bound, and on the right demonstrates the results of androgen deprivation therapy. While this treatment is effective for most tumors, after approximately 2 years of therapy, the tumors become castration-resistant, which means that the cancerous cells have mutated such that they are no longer androgen receptor dependent, so that even with very low levels of androgen in the body, the cancerous cells are still proliferating. There are a lot of reasons as to how these AR are becoming reactivated, like gene amplification or alternative splicing, and possibly many other factors that we still haven't figured out yet, but I want to focus my presentation today on mutations. Specifically, 2 mutations that occur at the ligand binding domain in such high frequencies of clinical samples that they are termed hotspot mutations. Hotspot mutations are defined as a mutation that occurs in a set of tumor samples significantly more frequently than expected from a background frequency, characterized by genes, cancer types, mutation types, and sequence contexts. So looking at this graphic, the y-axis denotes the frequency of the mutation, and the x-axis denotes the location and the type of mutation. The 2 mutations of interest to us are the T878A and the W742C mutations, which are both located on the ligand binding domain and represent one of the highest frequencies of occurrence along the androgen receptor. One of the more critical challenges of oncogenomics is to distinguish the mutations that confer tumorigenesis, aka the driving growth of the cancer, as opposed to those that provide no selective advantage to tumor growth, but rather occur stochastically or randomly. But what implicates this challenge even more is the phenomena of pairs of hotspot mutations that frequently occur together in the same clinical sample, and the potential that this co-mutational pattern may be prevalent because there may be a synergistic relationship between the two that makes it more successful at driving the growth of the castrate-resistant prostate cancer. Our goal was to recreate the different scenarios that presented in clinical samples of wild-type, single mutation, and double mutation AR using a synthetic androgen R1881 and clinical inhibitors docetaxel, DTX, and enzalutamide, MDV, to measure and compare the relative activity levels of each AR. The wild-type androgen receptor is our control and proves that with the androgen inhibitors, the levels of AR activity is less than the R1881 administered by itself, but still less than the control, which had no added androgens. Consequent experimental data with each mutant singularly shows that the activity levels of AR are marginally higher than its wild-type counterparts, but the blue control column is still relatively the same. But when we get to the double mutant, ARDM, the activity levels of the AR is increased exponentially. If we look closely at the graph, we can see that even the blue control column is almost 30 times more active than the other control columns, as well as being more than twice as active as the other red columns. This extremely high level of AR activity, even when treated with androgen inhibitors, may be a clue towards the mechanisms of castration-resistant prostate cancer, the type that tends to reoccur after 2 years of successful androgen deprivation therapy. The same conclusion can be reached with immunofluorescent staining, where the AR with both mutations present much brighter stains than the AR with just a singular T878A mutation. But the condition of the double mutation is one that is contrived by lab conditions, and we needed to prove that this co-occurrence happens naturally at a significant frequency greater than the chance in the wild-type androgen receptor of clinical samples of prostate cancer. So through the CBio portal for cancer genomics, I was able to procure a database of information on the types of mutations, protein changes, and study and sample IDs. From this database, I extracted the relevant data for the T878A and W742C mutations. Using a co-occurrence analysis as a framework, we found the statistical significance of co-occurrence among hotspot mutations that arose together in individual tumors. The implications of this finding demonstrates the need for more precision oncology and genomic mapping, because once we can profile the molecular sequence of tumors, target specific alterations and combinations of alterations during therapy, we may finally be able to combat the treatment-associated resistance mutations that will prolong life expectancy and quality of life in our patients.